Hello boys, welcome back to another session of video tutorials. This session is meant for class 12th students and this is meant for 13th August 2020. Our memory helps us that we have successfully completed principles of inheritance and variation, and I hope you have understood all the intricacies of genes and how important they play in our life, and you also understood the pedigree analysis that will help you to understand why particular diseases happen in a family and what will be the reasons and what will be the future, all this. Now with that in our background, now we go back or we move forward with regards to why the genes are responsible for our characters. And if you analyze, the genes are present in a very great molecule called DNA. So we are going to discuss about DNA and RNA, nucleic acids. So we have entered into another new interesting chapter called molecular basis of inheritance. And as all of you know, in your junior classes, there are two nucleic acids. One is DNA, deoxyribonucleic acid. DNA, very important. This is our secret of our life. And second one is RNA, ribulose nucleic acid, which is very essential for preparation of proteins. And proteins are again what we are. Whatever proteins you have in your body, that shows what you are. So both DNA and RNA are made up of what we call nucleotides. So we say, if someone asks you what is DNA or what is RNA, you need to say that they are polynucleotidal chain. So you can write it as polynucleotidal chain. And both DNA and RNA consists of one sugar molecule, I am writing sugar, yes, one phosphate group, okay, and two nitrogen bases. So you can remember it as SPN. Both DNA and RNA are made up of three molecules: sugar molecule and phosphate group and nitrogenous bases. So this nitrogenous bases are of two groups, as you are seeing, purines and pyrimidines. As you are seeing in the diagram, you know that purines are double ringed. See, ring number 1, ring number 2, double ringed. Pyrimidines are single ringed. It has got only one ring. Remember, you have studied in chemistry, benzene, cyclic compounds. It's something like that. Okay. So you can also see that purines are having this carbon number also. So as pyrimidines. But the main difference between purine and pyrimidine is purines are double ringed structures, pyrimidine is single ring. And purines are of two types, as you are seeing. Adenine, it is abbreviated as A, and guanine, G. So adenine and guanine. And pyrimidine is of three types: cytosine, uracil and thymine. And in this you must remember that uracil is present in RNA alone. Uracil is seen only in RNA in place of thymine, whereas thymine and cytosine are present in DNA. So uracil is unique, remember this. And we talked about nitrogen bases. So there are two types, purines and pyrimidines. Purines are adenine and guanine. How to remember? You can remember it as PAG, P-A-G. Purines, adenine and guanine. Or reverse, you can remember GAP for the sake of exam. And pyrimidines, you can remember it as PY because there we wrote for purines P. So It is PIC2U or PIC-U2, Union Territory, PIC-U2. Pyrimidines, cytosine, uracil, thymine. And we are left with now one more sugar. Sugar, it is pentose sugar. 5 carbons, as you are seeing, 5 carbon chain will be there. And you can see it is just like we are having glucose, fructose, sucrose, like that. Here it is ribulose. That's why the very title is deoxyribulose, and R stands for ribulose. So this is ribose or ribulose sugar. And as you are saying, you can see carbon 1, 2, 3, 4, 5. So it's a pentose. So 5 carbon chain. And at second carbon, if you see, in place of hydroxyl, if O is missing, only H is present. Since O is missing, we say deoxy. D means minus of oxygen. Hence deoxyribulose. So this is DNA deoxyribulose, RNA ribulose. I hope you understood the difference. Now what is that you have to do? Draw the diagram immediately. Deoxyribulose structure, ribulose structure, and all these purines and pyrimidines. All the diagrams you need to draw on a paper and post it to me in our WhatsApp group. And when the sugar, it's a pentose sugar, nitrogen, as you are seeing, either double ring or single ring, and the combination of this, that is sugar plus nitrogen bases, they result in formation of what we call nucleoside. These are nucleosides, NS. Remember? NS. And it is easy to remember in the exam. How? Sugar and nitrogen bases. Sugar and nitrogen bases. So nucleoside, SIDE, nucleoside. And when this sugar and nitrogen bases, they join with the phosphate group, as you are seeing here. Look at this. That becomes nucleotide. So you got a sugar here, 5 carbon sugar. You got a nitrogen base, double ring structure, purine. And to this, phosphate is added. That means we can say S for sugar, plus P for phosphate, N for nitrogen base. So SPN is equal to nucleotide. What is the difference between nucleotide and nucleoside? This is the common question that will be asked. Nucleoside is sugar plus nitrogen base. Nucleotide is sugar plus phosphate plus N. So what is the difference between the two? Presence and absence of phosphate group, this one. And here you can see to one carbon nitrogen base is attached, and on the other side phosphate group is attached. So draw this diagram also. Then you know, when we say DNA is a polynucleotidal chain, RNA is a polynucleotidal chain. This is one structure. Like this, many, poly means many structures add together through a weak hydrogen bond. There is a lot of significance why that should be weak hydrogen bond, because of DNA replication. You will understand in future. So many nucleotides join together through weak hydrogen bond, making polynucleotidal chain. That becomes this. Now you can see here nucleotides. Now deoxyadenylate. Adenylate means adenine is there. That is why adenylate. Second, deoxyguanylate. Adenine, guanine acid. So this is the structure, guanine. And deoxythymidylate. Thymine is the structure. Deoxycytidine, cytosine is the structure. You can see here it is double ring structure. Here it is single ring structure. Similarly, deoxyribonucleates. These are all present in DNA. When we join together this, so there is something called base pair ruling. What is the base pair ruling? that is adenine combines with thymine and cytosine combines with always guanine. So it is AT at, and in between there will be double bond. So I can write adenine always pairs with thymine and vice versa. Guanine through triple bond, hydrogen bond, makes pairing with the cytosine and vice versa. And this will be the structure in case of DNA, deoxyribonucleic. What about RNA? In RNA, there will be, in place of T, U will be there, uracil, just now I said. So, ribonucleotides, you can see here, adenylate, that is adenine. Adenine is attached here, you are seeing here, this is adenine. And guanosine or guanolate, guanine is attached. And uridine, this is what I was telling you, this is uracil, uridine is attached. And cytolate, that is cytosine, is attached. So you have a lot of work to do today. Unless you draw this, you will not be able to understand. Once you understand this, then you know that genetic material, what is the genetic material in us and how important it is. And I also should tell you that this chapter is not only very important for the sake of CBSE exams. It is career making chapter. You can become a molecular biologist. And you know the importance of molecular biology given in COVID-19 situation. This will make you a very good scientist. So for that, the base is this chapter. And you must understand that in the exams that almost this chapter has got 8 marks, as the evolution chapter is deleted for your batch. So it is only genetics chapter and molecular basis, that is 15 marks. So focus on this, don't neglect it, draw the diagrams, practice well. You understand that it is very, very easy. This chapter will become very easy, and there will be a lot of interesting experiments that we will discuss in our next session to prove that DNA is the genetic material. And the DNA, a scientist by name Frederick Griffith, has proved by conducting so many experiments, very interesting experiments, that DNA is the genetic material. And this was supported by Avery, McCarthy and MacLeod. 3 scientists together supported in saying that DNA is the genetic material. And we must be thankful to another 2 scientists in 1952, Alfred Hershey and Martha Chase. It's famously known as Hershey and Chase experiment, which proves beyond doubt that DNA is the only genetic material. So to understand all these experiments, of course, we must appreciate that, we must be thankful to 2 great scientists, Watson and Crick. Watson and Crick, Professor Watson and Crick. These 2 scientists made to the world its a structure, double helix structure, Watson and Crick, Professor Crick. And these 2 scientists made this world to know what the DNA is, and they proposed first time double helix structure, and because of the double helix structure, life has become easy for us. Even now we are in a position to cure genetic diseases that we discussed in our previous chapter. So basically today what I am trying to cover is, it is the structure of DNA. If we can make the structure of DNA by practicing all these diagrams, make 2 nucleotides connect with each other, that itself is a gene. And gene is the secret. If someone asks you what is your secret, you will say genes are our secrets. And in this chapter, at the end of the chapter, you understand that it is the protein